Okay, so welcome back. We have been discussing on the embryological evidences of organic evolution, and we saw various structures among the different groups of organisms that could provide the evidence for organic evolution. We also saw the four postulates put forward by von Baer, and here we'll be discussing on the Haeckel's recapitulation theory. Actually, von Baer's rule could be interpreted in such a way that similarities between embryos could be due to inheritance from common ancestry. That is, embryos exhibit homologous similarities just as adults do. However, later in the 19th century, exactly in 1905, Ernst Haeckel, he's another German embryologist, he suggested that embryos of higher animals repeated the adult stages of their ancestors. And this particular concept was put forward as the theory of recapitulation or the recapitulation theory, or it is commonly known as the biogenetic law. Haeckel's biogenetic law or Haeckel's theory of recapitulation. So here, the different stages in the development of an organism from zygote to adult, it is known as the ontogen. Okay, that is where we looking. So biogenetic law of recapitulation, or simply the theory of recapitulation, it was actually Ernst Haeckel. He published volumes, a book in two volumes in 1866, and the book was called General Morphologie der Organismen, that is general morphology of organisms. And in that he formulated a theory of the biogenetic law of recapitulation. And he put forward the concept of ontogeny recapitulates phylogeny. Ontogeny means the development of an organism or the different stages in the development of an organism from zygote to adult. And these what you call different stages are collectively called as ontogeny. That is development of an organism. Okay. Now phylogeny, it is the evolution of a species uh, from another species, or it is actually an evolution of species, development of a species. Okay, so phylogeny refer to the evolutionary history of a species, uh, the complete set of changes happening during the development of a species. It is what is referred as a phylogeny. So ontogeny, the development of a individual; phylogeny, development of a species. Okay, ontogeny, it happens within the lifespan of an individual, while phylogeny, it takes millions of years. Okay. So it says that ontogeny recapitulates. It is simply the repetition. So whatever is seen during the embryonic development of an individual is actually a repetition of whatever has happened during the uh, evolution of a species. That is what the particular sentence referred to. Okay. Now, um, an organism during its development repeats its ancestral history. Okay. That could be the summary of the whole concept. So Haeckel's theory um, exerts on the concept that uh, embryonic stages of a higher animal resembles the adult stages of its ancestors, and not the embryonic stages of ancestor as was put forward by von Baer. Okay. So several examples are cited from embryology to support the Haeckel's recapitulation theory. So this is those are these are the ones which had been cited to support the recapitulation theory. Okay. The first one, the comparison of embryonic stages. Development of every organism begins from a single cell, that is a zygote. This zygote undergoes cleavage, produces a multicellular embryo. It passes through different stages like blastula, gastrula, etc. And all these are almost similar among different groups of organisms. And in general organization, especially, it could be compared to the lower forms. Okay, so according to recapitulation theory, the zygote represents the protozoan stage. The blastula represents the protozoans, colonial protozoans like volvox and like that. Then gastrula, it represents the coelenterate stage in the evolutionary history of the particular species to which it belongs to. Okay, so that is what the uh, comparison has been put forward by Ernst Haeckel. Okay. Now, the tadpole and the caterpillar. So in the life history of frog, we can find the tadpole stage, isn't that the larval stage, which is actually fish-like in form and character, having a laterally compressed tail, gills, and lateral sense organs. So the vascular system also bear similarity to a fish. So here the evolutionists describe that tadpole stage represents the descent of amphibians from fish-like ancestor. Okay, so tadpole actually indicates that the amphibians have evolved from a fish-like ancestor. Similarly, the occurrence of a worm-like caterpillar. So this worm-like caterpillar stage in the life history of a butterfly indicates that the butterflies has evolved from an annelidan ancestor. Okay, so I hope it is clear. Then the next example is branchial grooves and pharyngeal pouches. In the fish embryo, the gill cleft develop as series of pouches on the wall of pharynx. So on the pharyngeal wall, small pouches develop, and that later develops into the gill clefts. So these pouches are known as pharyngeal pouches or visceral pouches. Okay, corresponding to these pharyngeal pouches, certain grooves are developed on the surface of the pharynx, and they are known as branchial grooves. The branchial grooves on the outside finally meet the corresponding pharyngeal pouches from inside, and a perforation then develops, converting the groove into gill slit, opening directly from the pharynx to the exterior of the body. So I hope it is clear. Okay. On the pharyngeal wall, from inside, pharyngeal pouches develop, and from outside, corresponding to these pouches, branchial grooves develop. These pouches and grooves meet together and then develop into smaller gill cleft or gill slit. Okay, and these gill slits actually opens the pharynx to the exterior of the body. So as water passes through the gill cleft, it bathes the filamentous and vascular gills found on the walls of the cleft. Okay. So exchange of gases take place between the blood in the gills and water passing through the gill slit. Okay. So this is how the fishes breathe. Right. So fishes, they need these structures for respiration. But during the course of study of the embryonic stages of various higher vertebrates, we can see similar branchial grooves and pharyngeal pouches present, especially in the embryonic stages. Okay, so why such kind of structures are found when these structures are no longer of any function in the higher vertebrates? So the possible explanation could be that the repetition of ancestral character. Probably the ancestral fish inherited this embryonic pattern from still more remote ancestors in which the gill slits served more for food collection than for respiration. Okay, so this like structures actually shows that the ancestral characters are repeated during the uh, embryonic development or the embryonic development of a particular individual. Okay. The next example that supports the recapitulation theory is the aortic arches. All vertebrate embryos develop six pairs of aortic arches, which we have already seen in one of the previous presentations, and some of them later degenerate in the course of development in the higher classes of vertebrates. Uh, in mammals, like all the six pairs are developed in the embryos of birds and mammals, even though in the adults some of these pairs are missing or disappear. Um, the reason could be that the birds and mammals may have descended from an ancestor which had six pairs of aortic arches, and their embryos may be repeating those ancestral conditions. Okay, so the lower vertebrates do have six pairs of aortic arches in the embryonic stage, and in coming to fishes and amphibians and all, these six aortic arches may be present, but coming to higher vertebrates, what happens is some of them get disappeared during the course of development. Okay, the adult vertebrates, especially those of reptiles, birds, and mammals, do have only 3 or 4 aortic arches. Okay, but the embryonic stages of all these vertebrate groups do possess all the 6 aortic arches, and this may be uh, like explained that the these vertebrate groups may have descended from an ancestor having all the 6 aortic arches. Okay. Now coming to the development of kidney. 3 types of kidneys occur among vertebrates, and they are referred as pronephros, mesonephros, and metanephros. The pronephric kidney, it is the most primitive. It is found in the most primitive vertebrates. In mammalian embryos, we can see that it is a higher type, that is metanephric, while mesonephric it is found in the adult fishes and amphibians, and also in the embryos of reptiles, birds, and mammals. But adult higher vertebrates possess only the metanephric. So here also it could be like the repetition of the uh, characters of the adult lower groups found in the higher like the embryonic stages. Okay. Now we have the larval forms, the trochophore and nauplius, especially. So, like just like the embryos showing similarities, certain larvae of animals also have show resemblances. Okay. For example, trochophore, it is a larva for a wide variety of invertebrates like platyhelminthes, nematodes, annelids, mollusks, and bryozoans. This indicates that the ancestors of all these animals looked like or resembled trochophore larvae. So, by producing a trochophore, the present day form are only repeating the ancestral character. Okay. Similarly, Nauplius is a larval stage occurring in the development of all the crustaceans. Okay. Uh, and again, this could also be pointing to the this recapitulation of the characters of the lower forms. Okay. Now, regarding the vestigial organs, the development of some non-functional structures in some embryos, which are to disappear later, can also be explained in the light of recapitulation theory. For example, the canine teeth appear in the embryonic stages of rabbits, which disappear before birth. Okay, so after birth, they don't have any canines, so they have a gap at that particular space, at that particular region, which is known as a diastema. But in the embryonic stages, they do have canine teeth. So similarly, the posterior pair of limbs appear in the embryos of whales, but later, before birth, it becomes vestigial. So this also like reflects a recapitulation theory, supports a recapitulation theory. Now, what are the criticisms and present status of the recapitulation theory? So, unlike Haeckel's view, von Bayer's rule states that the embryos of higher animals resemble only the embryos and not the adults of lower animals. Uh, and this was supported by many like people like Gaston and Debier. And according to Gaston and Debier, ontogenies of animals, that is the embryonic developmental stages. Okay, ontogenies of animals related by descent, animals that are related by descent, still remain similar up to a certain point, but then diverges. So therefore, embryological stages do not resemble the adult ancestors, but only the embryonic stages of the ancestors. Okay. So here it says that embryonic stages of higher animals resemble only the embryonic stages of ancestors and never the adults. Another point was put forward by Russian studies conducted by Russian biologists, and according to them, the developmental stages of an animal may resemble the adult ancestors or the embryonic stages of the ancestors, or may deviate somewhat from either of this, or may bear no resemblance at all. So, what we can summarize is that relation between ontogeny and phylogeny does not follow any set of rules. Okay, this was formulated by series of and they established that during development of embryos, during development of embryos, they may at certain
like structures or events to those of the ancestral